Hello, everybody. It's my pleasure to introduce everybody to another talk in the series of the 2023 Global Immunotalks. My name's Laura McKay. I'm an immunologist from the Doherty Institute at the University of Melbourne. And on behalf of all the 2023 global organizers, I would like to introduce this week's speaker, Dr. Taras Kreslavsky from the Karolinska Institute in Sweden. So Taras is originally from Russia. Following his undergraduate training in St. Petersburg, he moved to Harvard University to carry out a PhD with Harold von Boehmer. And then following a postdoctoral stint in the US, Taras then moved to the laboratory of Meinrad Busslinger at the IMP in Vienna, before then in 2018 starting his own independent laboratory at the Karolinska, where his group is today and where he's currently an assistant professor. So Taras is well known for his work on the transcriptional regulation and fate decisions of various innate-like lymphocyte lineages. He's a member of IMGEM, the Immunological Genome Project Consortium, and he combines cellular, molecular, and systems immunology to look at cell fate decisions. Now, his laboratory currently is working on two different avenues, looking at cell fate decisions of B cells and also various innate-like lymphocytes. And we'll hear about both of those later in your talk, Taras, and we're so excited for that. But before we get into your science, first, I think the audience would love to get to know you a little bit better. And so as part of our tradition here at Global Immunotalks, we'd love to ask you a question. And considering that you're quite a recent independent lab head, we would love to hear a little bit more about that. And particularly, what's been the most impactful decision that you've made so far, which has really impacted your scientific career?
Yeah, Laura, many thanks for this very kind introduction. And yeah, I think one of the most important decisions that we made 5 years ago when I was starting the lab was to invest in two different types of projects. On one hand, we had the preliminary data and some projects that could sort of naturally stemming from my postdoctoral work. And on the other hand, we decided to use some of our limited resources to invest into completely different type of projects where we didn't have any preliminary data, but we had exciting questions. And I think by having this combination, and I will be giving you some examples of these two project types in the course of my presentation today, I think made this 5 years extreme fun. And maybe here I will start sharing my screen. And wait a second. Does it work? Can you see the slides?
Yep, that's great. Thank you so much for your advice, and we're so looking forward to your talk. Please take it away.
Thanks. So I will start first with the first type of projects, which directly stems by my postdoctoral work with Meinrad Busslinger in Vienna, and that are on cell fate decisions that lead to formation of B-cell memory. And then I will switch the gears completely, and in the second of my talk, describe very preliminary results on our second passion, on the biology of gamma delta T cells. But let's start with the B-cell memory. So a quick reminder for those of you that don't remember that upon activation of naive B cells with cognate antigen, and if these B cells receive certain T cell help, they undergo a burst of proliferation and enter a tripotent state that is often referred to as activated precursors. And these activated precursors can either immediately make plasma cells, and this would be plasma cells that most likely are secreting antibodies of low affinity, but they can be generated very, very quickly in the response, and therefore provide the first line of protection. On the other hand, some of these activated precursors can enter germinal center reaction in an attempt to improve the affinity of the antibodies. And this is a process where B cells induce a very specialized molecular program and introduce mutations in their antibody genes, compete for antigen and T cell help, and in this iterative process, with each cycle of the process can potentially improve the affinity of the antibodies to antigen. And eventually, germinal center B cells can differentiate into either plasma cells that will home to the bone marrow and would secrete the antibodies that can then protect us if the same pathogen comes back, or they can differentiate into memory B cells, a diverse pool of which is supposed to be there, particularly useful in case we encounter a new variant of the pathogen, because maybe one of these diverse memory B cells would be able to recognize this new variant. And then finally, there is a long-standing notion that some of these activated precursors would directly bypassing the germinal center reaction and be directly contributing to the memory cell pool. But this early memory B cell biology was not very well understood. It was not clear what exactly are the signals that drive their differentiation. It was not clear what was the contribution of these cells to overall memory cell pool left behind at the end of the response. And we, for the reasons that I don't have time to discuss today, were very interested in this cell-fate decisions that activated precursors make in the first days of the response. And we decided to dissect them by single-cell RNA sequencing. And to this end, of course, this is early in the response. We need to get sufficient cell numbers. The clonal expansion had just started. So to get enough cells, we can use the following trick. We can take B cells with known antigenic specificity. In this case, B1A B cells from B1A high mice that recognize a hapten called nitrophenyl and P. We can transfer them into congenically distinguishable recipients, and then we can immunize these recipients with this model antigen and see what happens to the cells early in the response. And if we do this exercise around day 4 after immunization, we can start seeing the first BCL6 positive germinal center B cells. By that time point, we already have some of this IRF high early plasma cells. So we reasoned that if we sequence the cells around day 4 after immunization and spike in some cells from an earlier time point, day 3.5, we should be able to capture all developmental transitions of the activated precursors into the 3 populations. And when we did that and got the sequences back, the result looked from the first glance quite boring, because we could find indeed a small population of emerging germinal center B cells, a small population of plasma cells, and a large group of cells that we thought correspond to the activated precursors. So it looked like we just paid a lot of money to find back what was already known in literature and what we just saw by flow cytometry. We can see activated precursors differentiating into germinal center B cells and early plasma cells. And the only puzzling thing here was that we couldn't immediately pinpoint cells that would correspond to early memory B cells. But we thought that, okay, maybe this is such a minor differentiation pathway that we just didn't sequence enough cells to find them back. However, when we started looking into this data set further, we realized that maybe there is something interesting here. Because, for example, when we used RNA velocity, and this is an approach that tries to predict the future transcriptional state of the cell by looking at this ratio between spliced and unspliced transcripts, RNA velocity suggested that maybe all 3 populations are diverging from each other. And that's what you expect for plasma cells and germinal center B cells diverging from activated precursors. But this is not necessarily consistent with the idea of that this large population here correspond to the activated precursors at this point. And we started to think that maybe we are dealing with a situation like this. And maybe at this very early time point, a lot of cells already made the lineage choices, and we look at the 3 lineages that are largely separated from each other. And indeed, if we look at the same data set in a different embedding here, and then add cells from day 2.5 after immunization, we nicely fill the lacunes between the populations, suggesting that maybe indeed day 3.5 is already very late for this response. And what convinced us that there is some biological reality behind this bioinformatical analysis is behavioral cell cycle related genes. Because if we look at the cell cycle phase distribution in these populations, we can see that germinal center B cells are very proliferative. But this large population here seems to be largely quiescent, largely in G0 or G1 phase of cell cycle. And there are 2 possibilities here. Either we are dealing with the cells that are just being recruited to the response, and naive cells are just starting to get to divide, or we are dealing with a population that is actually exiting this cell cycle after proliferation. And to discriminate between these possibilities, we can make a simple experiment. We can label this naive B cells before transferring to recipients with a dye that will be diluted during proliferation, so trace violet, and then see whether the cells got divided. And indeed, if we do this exercise, you can nicely see that all antigen-specific B cells here had divided many times, diluted the cell trace violet to the completion, indicating that all the cells just underwent a very drastic burst of proliferation. And what we are dealing with here is exit from cell cycle. And indeed, if we look at Ki67 expression levels for a proliferation marker, you can see that at 2.5 activated precursors express high levels of Ki67, but then very rapidly they lose expression of Ki67, indicating their transition to quiescence. And what can be the cells that become quiescent after immunization? Obviously, it could be early memory B cells. And indeed, this population express some marker genes for memory. But of course, the definition of memory is not just the cells that cells are exiting from cell cycle, but it's their persistence after the response had ended. And to address the contribution of this pathway to the overall memory cell pool left behind at the end of the response, we next decided to perform a fate mapping experiment. We utilized the Cree line developed by Kurasaki's lab, S1PR2 Cree, and S1PR2 is a gene that is highly specifically expressed in germinal center B cells. And in this system, we can permanently label, by removing a stop cassette in front of the tomato, germinal center B cells and their progeny with the expression of tomato fluorescent protein. And if we do that with B1A B cells in the system that I just showed you, we used for single cell RNA sequencing, at day 4 after immunization, germinal center B cells will be already largely labeled with tomato expression. But if we look at 1 month later at the memory cell compartment, we can find some tomato positive cells, but a very large fraction of the cells that we're looking at are tomato negative, indicating that they were generated by this pathway. And we know that the cells proliferated like crazy, so it must be this pathway that generates the majority of memory B cells in the system. But of course, in this situation, we are dealing with relatively high naive precursor frequency because we transfer B cells with known antigen receptor. We have a relatively high initial affinity of BCR to antigen, and of course, these parameters can change the response. So next, we decided to check what happens in normal polyclonal situation. We use the same immunogen and P conjugated to OVA, and then we looked at antigen-specific B cells at different time points of the response. And we saw exactly the same thing. We saw that, yes, we have a little bit more of tomato positive memory, but still a very large fraction of memory cells are tomato negative, indicating that they're generated by this pathway. So not only a very large fraction, if not the majority of activated precursors exit cell cycle early in the response to become early memory cells, but this early memory then can make a very large contribution to overall memory cell pool that is left behind at the end of the response. Now, how conserved is this pathway across different settings and different species? To start addressing this question, we first collaborated with Davide Anghiletti's lab in Gothenburg, and Davide studies influenza A infection in mice. So he infected the mice with this virus and then looked at antigen-specific B cells at day 7 after immunization. And when he did that, he can find back a large population of cells that were neither germinal center B cells because they didn't express BCL6 nor plasma cells because they didn't express RF4. And these cells are exactly the cells that start to downregulate Ki67, as you can see here on this heat map. And if we look at this, the markers of the cells that would express some of the memory cell markers, such as CCR6. So at least in this infection model, we seem to see the same picture. Some of the cells exit the cell cycle early in the response to this, to make this early memory B cells. Now, is this conserved across species? To address that, we collaborated with our neighbors here on the floor with Karen Lares group, and Sebastian Ols and her lab were performing RSV nanoparticle vaccination in macaques. And when he looked at draining lymph nodes after vaccination early in the response at day 7, again, we get an antigen-specific B cells that clearly had expanded. And then we look at the distribution of the populations, and we see a small population of germinal center B cells, a small population of early plasma cells, and then a large group of cells that are neither of those and that downregulate Ki67 and that express memory cell markers such as CCR6 and CD95 and CD27 that I'm not showing you here, indicating that this pathway is conserved seems to be conserved in primates as well. The next question that we asked ourselves was what is the scale of the transcriptional changes that take place in activated precursors when they differentiate into the 3 lineages? And if we compare activated precursors early in the response to their contemporaneous germinal center B cells and plasma cells, perhaps not surprisingly, we see that transcriptomes of these 2 affected populations diverge very rapidly from that of the common activated precursors. But now, if we look later in the response and focus on memory B cells, If we remove cell cycle-related genes from the analysis, they cluster very closely together with activated precursors, although now we're comparing day 2.5 with day 50 after immunization, indicating that in the first approximation, early memory B-cell differentiation is nothing else but cell cycle exit of activated precursors. It doesn't look like there are major transcriptional changes otherwise in the cells. So it looks like activated precursors early in the response lose some mitogenic signal, and therefore they differentiate into this early memory B cells. What can be this mitogenic signal? Here from literature, one can get a very good idea that it might be antigen availability, because, and this is in line with publications by many groups, early after immunization, and I'm showing you here immunization of macaques with fluorescently labeled vaccine, but we see similar things in mice. You can see that antigen is broadly available in the draining lymph nodes, suggesting that every B cell that is antigen specific in this lymph node would have access to it. But then a couple of days later, at day 3 after immunization, all the antigen is concentrated on germinal center B cells, all the fluorescent signal in germinal centers, all the fluorescent signal colocalized with the markers of the germinal centers, indicating that the cells that landed outside of the germinal centers are likely to lose contact with antigen, or at least have a strongly decreased contact to antigen. So can that be the reason why the cells are, why this early memory B cells are exiting cell cycle? To address this, we can come back to the mouse system and use a simple trick. We can transfer again antigen-specific B cells, immunize the mice, and then at day 3.5, when we start seeing the first signs of cell cycle exit, we can try to provide extra antigen and see what happens to the B cells in this case. And if we do this exercise, we can nicely see that the activated precursors are starting to continuing their proliferation if we provide extra antigen, as judged here by EDU incorporation, and that indicates that antigen is indeed limiting here, and by provision of more antigen, we can re-recruit the cells into the cell cycle. But moreover, we also saw a rather drastic wave of plasma cell differentiation from the activated precursors when we provided extra antigen. And I think that actually can give one of the reasons why we have this, what is the function of this early memory B cells. Because we always think about memory function in the secondary immune responses. But here we have a situation where we have a primary immune response. And imagine that, so we preserved excess of early activated precursors in a quiescent state, so they're energetically cheap. And then let's say we have an infection situation, and the failure to contain the infection was all of a sudden manifested by increased antigen availability. Then we can rapidly convert this cheap early memory B cells into energetically expensive plasma cells and attempt to fight off the infection through that mechanism. At least that's a tempting hypothesis here. So what I showed you so far, and this is all published results, we now move to the unpublished data, is that at least in some settings, a very large fraction of the activated precursors undergo some sort of differentiation by default into early memory B cells. And these early memory B cells can then make a very prominent contribution to overall memory cell pool that is left behind at the end of the response. And these events are largely driven by antigen availability that becomes very limited outside of the germinal centers after the first few days of the response. And now we're trying to understand what can be the reason of existence of these early memory B cells, what can be their functional roles that would be unique to them and not to germinal center derived memory B cells. We have very early in this path, and I decided not to include any data slides on that. But we think that at least in one of the settings, this is very preliminary, this may still fall apart, but at least in one of the settings, we seem to see that early memory B cells are potentially the most clonally diverse population, suggesting that they're more clonally diverse than germinal center derived counterparts, suggesting that if that holds true, they may be a repository of clones that were, some of which were not particularly useful in response to this specific antigen. But that can be nevertheless worth preserving in case we encounter new variants. And at the same time, we also see that in secondary immune responses, early memory B cells, unlike their germinal center derived counterparts, can very nicely enter the germinal center reaction and Therefore, they can further adapt their antigen receptor to the new challenges. And thinking along the same lines, we wanted to have a molecular comparison of early memory B cells and germinal center-derived memory B cells. And to this end, we performed again a single-cell RNA-seq experiment. In this case, we studied a polyclonal response to NP-OVA with fate mapping with the same S1PR2 tomato system. And then Vasilis Glaros, a very talented PhD student that works on this project, sorted separately tomato positive and tomato negative populations of B cells, hashed them with barcoded antibodies, and then we mixed them together and performed a single-cell RNA sequencing on these populations. And when we look at the clusters of memory B cells, of course, we also included germinal center B cells and plasma cells, but if we sort of gauge on the memory cell specific clusters, we can find here in blue are tomato negative cells, in red are tomato positive cells. And you can see that here at the lower part, you see more of blue dots. On the upper part, in the upper part, you have more red dots, but overall there are no unique clusters, there are no unique group of cells that would be exclusively tomato positive or tomato negative, suggesting that at least on the transcriptional level, participation in the germinal center reaction or early memory differentiation pathway do not change the transcriptomes of the cells dramatically in some deterministic fashion. But what was interesting in this data set that a large fraction of cells in the memory compartment expressed genes that normally are associated with marginal zone B cells. And you probably remember that marginal zone B cells is a lineage of B cells that is thought to differentiate split from conventional follicular B cells at the late stages of B cell development. And this differentiation is driven by notch signaling through notch ligands available in the marginal zone of the spleen. And these cells are thought to be more innate-like population that is there to provide the first line of defense against blood-borne pathogens. But here we're looking at the memory compartment, and we find the cells with the same signature. And we're not the first people who are seeing those. For example, Andreas Radbruch also observed this in his single-cell RNA sequencing datasets. But no one to our knowledge investigated what is the origin of the cells and what is the function of the cells. So we decided to try to address these questions. And first question we asked, are those marginal zone-like memory B cells germinal center derived or non-germinal center derived? And you can already see from the single-cell RNA sequencing data that here in this area we see both red and blue dots, suggesting that probably the cells can be generated by both pathways. And indeed, when Vasilis performed flow cytometric experiment with fate mapping, and if we look at tomato positive and tomato negative memory compartment and stain for the markers of marginal zone B cells, for example, high levels of CD21 expression, you can see that in both cases we see a very nice marginal zone population. Although reproducibly, we see more marginal zone phenotype cells in the early memory compartment, suggesting that both pathways can generate them, but maybe early memory B cells are more efficient in seeding this marginal zone memory niche. The second question we asked, okay, what about the origin? What are their naive precursors? Because if we think about the cells, about marginal zone B cells as a lineage, then we would expect that the cells should maintain their lineage identity in the course of immune response, and marginal zone naive B cells should generate marginal zone memory and vice versa. To address whether this is the case, Vasilis performed an experiment where he sorted naive cells with follicular marginal zone phenotype from this B18 high mouse where we know antigen specificity. And then he immunized the mice with our model antigen and looked at the memory compartment 1 month after that. And when he did that, we realized that it actually doesn't matter whether you start as naive marginal zone B cell or naive follicular B cell. At the end, you have a chance to generate both memory subsets, no matter what were your naive identity. Suggesting that maybe this view on marginal zone B cells as a stable lineage is sort of an oversimplification, and maybe it's more of a niche-induced state that can be induced either in naive B cells or in early memory B cells or in germinal center-derived memory B cells. Now, the next question, and here we are just in the beginning of the path that we wanted to address, what is the function of this marginal zone state of memory B cells? And one of the ideas that we got from literature about naive marginal zone cells was that maybe it's something to do with longevity, because the half-life times of marginal zone B cells are higher than that for follicular B cells. To see if marginal zone B cells, to see what is the longevity of this marginal zone like memory cells, Vasilis performed an experiment. Oh yeah, so basically we just showed that there is a complete plasticity between these two populations in the course of the immune response. So to test what is the longevity of these populations, Vasilis performed an experiment where he immunized the mice, transferred B1-8 high B cells, immunized the mice, and then performed fate mapping and analyzed them in the course of the whole immune response. And then he looked at the memory cell compartment over time. And what became very clear was that in terms of frequencies, the marginal zone B cells accumulate over time quite dramatically. And Vasilis, I think, is now at the fax machine acquiring the day 150. So we'll see if the numbers go even higher. And if we stratify this memory compartment by tomato expression for germinal center derived and non-germinal center derived cells, we can nicely see that in both populations, marginal zone phenotype cells increase in their frequency, but the kinetics are somewhat different. It seems to take longer time for germinal center memory B cells. Now, this is not because the cells increase in numbers, because if we look at absolute numbers of the cells, it seems like the numbers of the marginal zone memory phenotype cells here in green is extremely stable over time. And what is happening is contraction of the follicular phenotype memory cells, suggesting that maybe this marginal zone state allows for long-term survival of memory B cells, and potentially marginal zone of the spleen might provide a specialized niche for long-term survival of memory B cells. At least that's the hypothesis that we're currently addressing. What we don't know here is how much of the exchange between the two compartments is, how much of the conversion of follicular B cells into marginal zone memory B cells takes place, and the other way around. And finally, after seeing this rather drastic accumulation of memory B cells after immunization, we decided to revisit a cross-species controversy that exists in marginal zone B cell field, because in mice, marginal zone B cells are thought to be mostly naive B cells, although there are some reports before us about marginal zone-like memory. But in humans, a lot of cells with marginal zone signatures seem to be antigen experienced. And we reasoned that maybe one of the differences between the situation in mice and humans is exposure to antigens, because we mostly work with SPF mice. And therefore, we collaborated with Stefan Rosshart and Jonathan Caquette and looked into wildling mice that are black 6 mice that were reconstituted with basically wild-type microbiota. And when we look into this dirty mice, we can nicely see that in marginal zone compartment, you have increase in the frequency of cells with memory phenotype. And these are the markers that were defined by Max Slomczyk's group that are actually mostly marking germinal center derived memory cells, suggesting that we're probably underestimating the frequency of memory cells in marginal zone compartment. And if we look at the quantification here, you see that the increase is true for marginal zone B cells, although of course we see also an increase in memory cells in follicular compartment as well, indicating that maybe at least in part this cross-species difference is related to the exposure to microbiota and pathogens. So what I showed you in this part of my talk is that in addition to this layer of memory B cell identity in terms of the ontogeny from either germinal center B cells or directly from activated precursors, there is a second layer of identity, and some of the memory B cells from both pathways acquire a marginal zone phenotype, and this marginal zone phenotype seems to be associated with rather extreme longevity of the cells. And we're now trying to test why the cells exist by genetic ablation of this population. And our current hypotheses are that on one hand, maybe marginal zone of the spleen provides some unique survival niche for memory B cells. On the other hand, it might be, of course, a strategic location for memory B cells to be for response to blood-borne pathogens. So now let me switch the gears completely and talk about our second passion, about gamma delta T cells. You probably know that in all vertebrates, from cartilage fishes to mammals, we have three classes of adaptive lymphocytes. We have B cells, we have alpha beta T cells, and we have gamma delta T cells. And for B cells and alpha beta T cells, we understand very well what are the principles of antigen recognition, what kind of antigens, what classes of antigens the antigen receptors can recognize. And because of that, we understand very well what the function of the cells is. But for gamma delta T cells, we know only a handful of antigens. We don't have any idea about the general principles, if they exist, for antigen recognition through these receptors. And because of that, I would argue we understand very poorly function of the cells. What we do know, however, that during the development, a lot of gamma delta T cells, although not all, induce effector programs. And these effector programs, at least some of them, seem to be induced because of agonist selection, because of recognition of endogenous ligands that are expressed in the thymus and result in the differentiation of these innate effector cells. In most of the cases, we have indirect evidence for that. In a few cases, we know the ligands that induce this agonist selection, and they fall into two categories. Either it's something MHC related, usually MHC1 related, and here the recognition involves the CDR3 loops of the TCR formed by VDJ recombination, although sometimes configuration of how this binding happens can be very, very strange. And it's usually very, very small subsets. So it's not clear whether there is any focusing of gamma delta TCR repertoire on MHC related molecules, or whether we have good tools. We expect a T cell to recognize an MHC, and there will find rare cells that actually do it. The second class of ligands that became very popular in the last years are butyrophilin-related molecules that function in a completely different mode. They function more or less like super antigens. Their interaction with germline encoded sequences in the gamma chain results in agonist selection or activation or accumulation in the tissues of gamma delta T cells utilizing particularly the gamma segment. Now, in general, if we look at the chain combination utilized by different gamma delta T cell subsets, we can realize that often a particular combination of gamma and delta chains results in differentiation or coincides with a particular effector program, particular tissue localization, and so on, again, sort of strengthening this notion that some of these gamma delta T cells should be selected on endogenous ligands. And these are the chain combinations that are very well described in literature. But if these days you have a lot of NGS data sets, and for example, there was a very nice paper from Thomas Winkler group with single cell RNA-seq, VDJ-seq of gamma delta T cells from various tissues. And if you look at the spleen, you can find some of these canonical, very well described subsets. But one of the most dominant chain combination is V gamma 4 with V delta 7. And we don't know anything about antigen receptor specificities of this TCR. And I will come back to this chain combination later. So we decided that we want to look in semi-unbiased fashion how predominant, how prominent is recognition of endogenous ligands by gamma delta TCRs, and we cloned about 40 gamma delta TCRs from mice and plugged them into reporter cells that have everything required for TCR signaling, that do not express their own TCR, and that have an NFAT reporter, fluorescent reporter that first shines in blue and then with time changes the fluorescence to red spectrum. But if we see on the FACS plot a fresh wave of blue like this, that means that the cell just underwent TCR signal. And then we co-culture the cells that are identical with everything except for the TCR that they express with a panel of cell lines and primary cells that we think correspond to what is present in the thymic environment, because thymus is where agonist selection in most of the cases takes place. And when we did this exercise, we could find about 8 or 9 TCRs, I'm showing you 7 here, that recognize something. For example, this TCR here clearly fires in response to something expressed by an endothelial cell line, but no other cell type activates it. This T cell receptor has a more broad pattern of reactivity. It also recognizes thymic fibroblasts and thymic double positive T cell lymphoma. and so on and so forth. And it looks like there are multiple different patterns of reactivity here, suggesting that not only the TCRs recognize endogenous ligands, but it should be rather diverse endogenous ligands, because different TCRs are activated by different cell lines. So is that recognition of the ligands that we already know, or at least that belong to the classes that we already know? To address this question, we can take advantage of the fact that a lot of our reactivities converged on a double positive T-cell lymphoma cell line that is called BCA. That actually makes biologically a lot of sense, because a lot of innate like lymphocytes are thought to be selected in the thymus by ligands expressed by double positive thymocytes rather than by thymic stroma. And here we have a transformed double positive thymocytes that activate our gamma delta TCRs. But we can also just take an advantage of this fact and see what kind of ligands this BCA cells express. And if we do RNA-seq on these cells, we can find that none of the butyrophilin-related molecules is expressed by this the cell line. A bunch of other molecules that were implicated in gamma delta TCR signaling are either not expressed or expressed at very low levels. But of course, not surprisingly, this thymocytes express some of the MHC1-related molecules. So could it be that all our reactivities are explained by something MHC1 related? To address this question, we can take advantage of the fact that these molecules should depend on their surface expression on beta-2 microglobulin, and we can just knock out with CRISPR-Cas beta-2 microglobulin from the cell line and see how the reactivity changes. And when we do this exercise, you can appreciate that only one of our TCRs lost the reactivity here, whereas all other TCRs maintain their reactivity to the BCA2M deficient BCA cells. And here we rapidly could figure out that this was a recognition of previously known gamma delta TCR ligands, non-classical MHC1 molecules T10 and T22. But most importantly, this result suggests that not only we have diverse recognition of diverse gamma delta TCR ligands, endogenous gamma delta TCR ligands by these TCRs, but also these ligands do not belong to known classes of gamma delta TCR ligands. So what are these molecules? And here we are again sort of in the beginning of the path. The screening approach that we use so far for ligand identification was cDNA library screen. And here we can take basically in bulk mRNA from ligand expressing cells, for example, from this double positive lymphoma cell line that activated a lot of our TCRs. And then we can clone it, the cDNA in bulk in an expression vector. And then we can create pools of cDNAs, of plasmids encoding this cDNAs by just diluting, titrating down E. coli. So we have roughly 200 clones per well of 96-well mini prep. They can then make this 96-well mini preps, transfect this pools into feeder cells. Here we use 293T cells, and then co-culture these feeder cells with our reporter cells. And if, and of course it's a big if, our ligand can be encoded by a single cDNA, then we should be able to see the reporter activation. And when we did this experiment, so far we found two molecules that look like a hit. Here is one, and I will not be talking about the second one yet. Here is a pool that clearly activated the reporter. Here on y-axis we plot the frequency of reporter positive cells. Each dot here is an individual pool, and it's clear that this something activates TCR in this specific well. And if we dilute this pool and then get to single cDNA, you can see that that there is very nice activation of the reporter cell lines when we transform this, transfect this cDNA encoding construct. And when we sequence this cDNA, to our surprise, we found something that you wouldn't expect to activate TCR signaling, because it's a cytokine receptor. IL-17 receptor A chain was the hit that was activating the reporter in our cells in this pool. But of course, this data by itself does not necessarily mean that this is because of the TCR specificity. To test this, Kiwi Ye, a very talented PhD student that works on this project, compared the TCR that we used in the screen, and this is a V-gamma-4 V-delta-7 TCR. You remember I was talking about the subset earlier that we found initially as a hit, with other TCRs that we had on our collection, and the majority of the TCRs were not activated by IL-17 receptor A, indicating that is TCR-mediated, TCR-specific reactivity. And at this point, I was pretty convinced that this is a typical situation in gamma delta TCR field, where you find a TCR that recognizes a molecule, and this is a single TCR that in universe you never find another one. But to our surprise, when Kiwi looked at our small collection of TCRs, there was a second TCR in our collection that also was activated by IL-17 receptor A. And this was also a β7 TCR, in this case with β1 chain. In both cases, we could completely abrogate the reactivity by introducing a lot of mutations in CDR3 delta region, but the reactivity was CDR3 delta dependent. And moreover, and that was nice to see, we could completely abrogate the reactivity by knocking out IL-17 receptor A from bigger cells, indicating that indeed this IL-17 receptor A was responsible for the activation of the TCR signaling when we performed these co-culture experiments with bigger cells. And finally, not only IL-17 receptor A was required to induce TCR signaling, but also it was sufficient to induce TCR signaling, because when we used recombinant protein, it very nicely activated the reporter. At this point, I was sure that next day we'll find we can use the IL-17 receptor A or the recombinant protein as staining reagent, find back in vivo the population that is recognizing this molecule. However, it took us about a year to establish a staining protocol, because tetramers do not work. You need to multimerize the IL-17 receptor A further. But finally, Kiwi succeeded in designing IL-17 receptor A multimers that stained our reporter cells that we know recognize IL-17 receptor A, that completely lose the mutants that do not recognize IL-17 receptor A, completely lose the staining. But I must say this, the staining, for reasons that we don't completely understand, is always incomplete. And that suggests that all the stainings that I will be showing you later is probably also an underestimation of the actual cell frequencies. So now, using this tool, Kiwi went to the ex vivo gamma delta T cells and stained them with the IL-17 receptor A multimers. And he saw rather huge populations of 1 to 3% of total gamma delta TCR repertoire in lymph node and spleen were stained with this multimer. And at this point, I thought, okay, this probably is not TCR mediated. It's too crazy that 1 in 30 gamma delta T cells would be recognizing a cytokine receptor. It doesn't make much sense. But Kiwi, to prove me wrong, next performed single cell sorts and then optimized single cell nested PCRs and sequencing of the TCR junctions. And when he sequenced a few cells from the single cell sorts that we knew bind the IL-17 receptor multimer, my view on this result changed completely, because all the cells had β7 had TCRs with β7 chain. And almost all of them were β4 β7, with one TCR being β1 β7. Strongly suggesting that we and this is exactly the β that we saw in the parental two TCRs, right? Strongly suggesting that we are probably looking at the IL-17 receptor A specific TCRs. Strange enough, we saw extremely diverse CDR3 regions in the cells, suggesting that CDR3 is somehow important, because we can mutate the CDR3 region and we can abrogate the reactivity in our original TCRs, but a lot of different sequences are allowed in CDR3 delta regions, and it's probably vδ7 somehow that is the most important determinant of this reactivity. And finally, to prove that these TCRs are actually IL-17 receptor A reactive, Kiwi cloned them and expressed in our reporter cells, and we tested all 7 TCRs that we have cloned so far, and all of them were very nicely reactive to recombinant IL-17 receptor A, formally proving that this large population of IL-17 receptor A binding gamma delta T cells in vivo are actually IL-17 receptor A reactive cells. Now, I told you this is a very, very early preliminary data, so we don't have the answer why we have the cells, why we have this reactivity, and what is the function of this interaction in vivo. What we do know, however, by looking at the surface phenotype of the cells, is that they express, if we get on IL-17 receptor A multimer stained cells, they express a bunch of markers that are associated with agonist selection, suggesting that they were selected on endogenous ligands, and we assume, but we're only importing the knockouts now, that maybe this is because they saw the IL-17 receptor A during their development in the thymus, and it looks like they from this marker expression, they might be inducing some interferon gamma secreting Th1-like program that we'll need still to show. So what I showed you in this part of my talk is that when we tested the reactivity of the gamma delta TCRs to endogenous ligands, out of almost 40 TCRs, we found about 8 that were activated by some of the cell lines or primary cells that we had on our panel. And we could identify ligands for 3 of these TCRs. In one case, it was the previously known MHC1 molecule. And for 2 TCRs, to our surprise, it was a cytokine receptor. It was IL-17 receptor A chain. And of course, it's very interesting to know why this reactivity exists. But interestingly, this was the reactivity of that was dominated by this V gamma 4 V delta 7 TCRs, and which make a rather sizable fraction of gamma delta T cells ex vivo. We don't think all of them are recognizing IL-17 receptor A, but probably a substantial part of this chain combination are recognizing the cytokine receptor. And from our data about the importance of V delta 7, about the wide array of sequences that I allowed in the CDR3 delta region, and the fact that we have at least 2 different gamma chains that can build such IL-17 receptor ATCR, it's tempting to speculate that they're dealing with a system that has some parallels with butyrophilins, but instead of pulling out of the repertoire and inducing agonist selection of gamma delta T cells that express particular V gamma chain, we are selecting TCRs by their V delta. And again, we need to figure out what can be the functional role of this interaction, and that's what we will be working on now. So these are the 2 stories that I wanted to share with you today. The key people in my lab that were responsible for the stories, for B-cell part, it's Vasilis Glaras, a very talented PhD student who was really driving this project with help from a postdoc in the lab, Annika Reinhardt. And for gamma delta T cell part, ex-postdoc in the lab, Josephine Dunst started this project, and then it was taken over by Kiwi Ye, another PhD student in the lab. And for B-cell projects, we collaborated quite a bit with my postdoctoral mentor, Meinrad Büßlinger, and René Rauschmeier in his lab. Karin Lore and Sebastian helped us with monkey experiments. Robert Manson and Charlotte Gustafsson helped us with bulk RNA sequencing. Jonathan Caquette and Stephen Roshardt in the University of Freiburg enabled the wildling experiments that I showed you briefly. And then Igor Adameyko, Artyom Artyomov, as well as also Björklund and Claudio Mirabella helped us a lot with bioinformatical analysis of the single cell RNA-seq data sets. Davide in his lab in Gothenburg helped us with influenza infection experiments. And Thomas Cray and Stephanie Curlin in his lab produced all recombinant proteins for the gamma delta TCR project. And Jan Kiselov provided the very valuable reporter cell line that we used in all these assays, as well as Beaker lymphoma cell line. And here are our funding sources. Thanks a lot for your attention, and thanks a lot for inviting me here.
Taras, thank you so much for delivering such a inspiring, fantastic seminar, such a wealth of data, and thanks again for presenting such new data. I'm sure we'll have so many questions from the audience for you. So just a little information on how to do that. So if we go to, from here, if we go to Twitter, and then there'll be a hashtag where you can search for the Global Immunospeaker. From there, you can find the tweet that says, ask questions for Dr. Taras Kreslavsky here, and then reply to that tweet, and Taras will kindly use his Twitter account to address all of your questions for quite some time. So thank you so much again for delivering such a fantastic talk for us today. And for everybody, please tune in next week. We'll have Jim Allison joining us. So very excited for that. And we'll see you all next time. Thank you so much.
Thanks.